Welcome to this scientific lecture on one of the most important discoveries in biology, how we determine the structure of DNA. This is a story of brilliant experimental work, careful reasoning, and rigorous hypothesis testing. In the early 1950s, scientists knew that DNA carried genetic information, but they did not know its three-dimensional structure. The key question was, how is DNA organized at the molecular level? You might ask, why does structure matter so much in biology? The answer is that structure determines function. To understand how DNA stores and transmits genetic information, we needed to know its precise molecular architecture. The primary experimental technique used to solve this puzzle was X-ray crystallography, specifically X-ray diffraction. This method allows us to probe the atomic structure of molecules. Let me explain how X-ray diffraction works and why it was so crucial for determining DNA structure. X-rays are directed at a purified DNA sample. When X-rays interact with the electrons in atoms, they scatter in specific directions. This scattered radiation produces a diffraction pattern on a photographic plate. The diffraction pattern is not a direct image of the molecule. Instead, it is a complex interference pattern that contains encoded information about the positions of atoms in the DNA molecule. The most famous X-ray diffraction image of DNA is Photo 51, taken by Rosalind Franklin and Raymond Gosling in 1952. This photograph provided crucial clues about DNA structure. Photo 51 showed a distinctive X-shaped pattern. This X-shape is characteristic of a helical structure. The spacing between the diffraction spots provided quantitative measurements of the helix dimensions. What does the X pattern tell us scientifically? The X indicates that DNA has a helical structure. The angle and spacing of the X arms tell us the pitch of the helix, which is the distance for one complete turn. From the diffraction data, Franklin calculated that the helix makes one complete turn every 3.4 nanometers. She also measured a repeating distance of 0.34 nanometers, suggesting regular stacking of bases perpendicular to the helix axis. Now let me explain how James Watson and Francis Crick used this data to build physical models of DNA structure. You might wonder, why build physical models instead of using computer simulations? In 1953, computers were primitive and could not handle complex three-dimensional molecular modeling. Physical models were the only practical option. Watson and Crick built models using metal plates for the bases and rods for the sugar phosphate backbone. These physical components could be arranged in 3 dimensions and tested against experimental data. Building models was not arbitrary. The models had to respect fundamental chemical constraints. This is where scientific rigor becomes absolutely critical. First constraint. Chemical bonds have specific angles and lengths that cannot be violated. Carbon-carbon bonds are approximately 0.154 nanometers long. Bond angles in organic molecules are determined by orbital hybridization. Second constraint. Erwin Chargaff had discovered that in DNA, the amount of adenine always equals the amount of thymine, and the amount of guanine always equals the amount of cytosine. This ratio is known as Chargaff's rules. The concentration of adenine equals the concentration of thymine, and the concentration of guanine equals the concentration of cytosine. What does this ratio suggest about DNA structure? It strongly hints at some kind of pairing relationship between A and T, and between G and C. This was a major clue. Third constraint. Bases can form hydrogen bonds, but only certain combinations are chemically favorable. Adenine can form 2 stable hydrogen bonds with thymine. Guanine can form 3 stable hydrogen bonds with cytosine. Here we see adenine and thymine forming 2 hydrogen bonds. The geometry of these molecules allows this specific pairing. Guanine and cytosine form 3 hydrogen bonds. This complementary base pairing is fundamental to DNA structure and function. The scientific method requires that we reject models that contradict experimental evidence. Watson and Crick initially considered several incorrect structures before arriving at the correct one. One early idea was a 3-stranded helix with the bases pointing outward. This model was quickly rejected because it did not match the X-ray diffraction data and violated chemical constraints. If the bases were on the outside, they could not form the stable hydrogen bonds required by Chargaff's rules. The model was chemically implausible. Even the great chemist Linus Pauling proposed an incorrect 3-stranded model in 1953. His model had the phosphate groups in the center. which was chemically impossible because the phosphates are negatively charged and would repel each other. Why do scientists make mistakes even with data available? Because interpreting complex data is difficult, and sometimes we are influenced by preconceptions. The scientific process corrects these errors through rigorous testing. Watson and Crick's breakthrough came when they realized the correct structure, a double helix with bases paired on the inside and the sugar phosphate backbone on the outside. The two sugar phosphate strands run in opposite directions, creating an anti-parallel double helix. The bases project inward and pair according to the rules A with T, G with C. This model explains all the experimental data simultaneously. It accounts for the X-ray diffraction pattern, Chargaff's rules, the diameter of the helix, and the regular spacing of bases. The diameter of the double helix is approximately 2 nanometers, consistent with X-ray measurements. There are approximately 10 base pairs per complete turn of the helix. Since one turn is 3.4 nanometers, each base pair is separated by 0.34 nanometers. A robust scientific model must be supported by multiple independent lines of evidence. Let me explain how the double helix model was verified. First, the model perfectly explains the X-ray diffraction patterns, including the helical pitch, the diameter, and the spacing between base pairs. Second, complementary base pairing naturally explains why adenine equals thymine and guanine equals cytosine. This is not a coincidence but a structural requirement. Third, and most importantly, the structure immediately suggests how DNA can be replicated. If you separate the two strands, each strand can serve as a template for building a new complementary strand. How does structure suggest function here? The complementary base pairing means that if you know the sequence of one strand, you automatically know the sequence of the other strand. This is perfect for copying genetic information. When DNA replicates, the two strands separate, and each serves as a template for a new complementary strand. This elegant mechanism is built into the structure ITS. Let me emphasize the iterative nature of the scientific process that led to this discovery. Step 1. Collect experimental data through X-ray diffraction. This provides quantitative measurements of molecular dimensions and symmetry. Step 2. Propose a structural model that is consistent with the data and respects chemical principles. Step 3. Test whether the model explains all available experimental observations, not just some of them. Step 4. Rigorously reject any model that contradicts even one piece of reliable experimental data. Step 5. Refine the model and repeat the process until you find a structure that explains all data with no contradictions. Why is this iterative approach so powerful? Because it minimizes the role of bias and speculation. Models that do not match reality are systematically eliminated, leaving only those that truly reflect nature. I want to emphasize the crucial role that chemical constraints played in determining the correct structure. X-ray diffraction data alone is often ambiguous. Many different structures could potentially produce similar diffraction patterns. Chemical knowledge constrains the possibilities. Bond angles restrict the possible geometries that atoms can adopt. You cannot arbitrarily place atoms anywhere. They must obey the laws of chemical bonding. The specificity of hydrogen bonding determines which bases can pair with each other. This is not arbitrary but follows from the electronic structure of the molecules. What would happen if we ignored chemical constraints and only used diffraction data? We would propose many impossible structures that could never exist in reality. Chemistry is essential for guiding us to the correct answer. Let me show you some of the quantitative relationships in the DNA double helix that had to be explained by the model. Let n be the number of base pairs in a DNA molecule. The total length of the DNA molecule is n times 0.34 nanometers, since each base pair adds 0.34 nanometers to the length. The number of complete helical turns is n divided by 10, because there are 10 base pairs per turn. The total twist angle is 36 times n degrees, since each base pair contributes 36 degrees of rotation. These mathematical relationships had to match the experimental measurements from X-ray diffraction. Any proposed model that gave different numbers would be rejected. The Watson-Crick model has been verified and extended by decades of subsequent research. Modern single-crystal X-ray crystallography has determined the exact positions of every atom in DNA with sub-angstrom precision. These structures completely confirm the Watson-Crick model. Nuclear magnetic resonance spectroscopy allows us to study DNA structure in solution, showing that the double helix is also the predominant form in aqueous environments, not just in crystals. We now know that DNA can adopt alternative helical conformations, such as A-form DNA and Z-form DNA, under certain conditions. But B-form DNA, the Watson and Crick structure, is the most common in cells. Let me conclude by highlighting the key methodological lessons from this discovery. Lesson 1. The most powerful scientific conclusions come from combining multiple independent types of evidence. X-ray data, chemical principles, and biological observations all supported the double helix. Lesson 2. Scientific models must make testable predictions that could, in principle, prove them wrong. The double helix made many specific predictions about DNA properties. Lesson 3. Proposed models must respect fundamental physical and chemical constraints. Creativity is important, but it must operate within the laws of nature. Lesson 4. Scientific progress often requires many iterations of hypothesis generation and testing. Watson and Crick tried many incorrect models before finding the right one. Lesson 5. In molecular biology, structure determines function. Understanding the structure of DNA immediately revealed how genetic information could be stored and replicated. Why does this discovery matter today? Because understanding DNA structure was the foundation for all of modern molecular biology, biotechnology, and genomics. We could not have sequenced the human genome or developed gene therapy without this fundamental knowledge. In summary, the determination of DNA structure exemplifies the scientific method at its best. We used rigorous experimental techniques, particularly X-ray crystallography, to collect quantitative data about molecular structure. We proposed models that were constrained by fundamental principles of chemistry and physics. We systematically rejected models that contradicted any piece of reliable experimental evidence. We iterated through multiple hypotheses until we found a single structure that explained all available data with no contradictions. This methodology remains the gold standard in structural biology today. We still use X-ray crystallography, now supplemented by cryo-electron microscopy and other techniques, to determine the structures of biological molecules. The principles established by this discovery continue to guide scientific research.